Hey everybody, welcome back to another video. This video is going to be part of a new playlist called Cell Physiology. It's really just going to cover some of the basic stuff that you have to understand about cell biology in order to really understand the larger human physiology picture. And if you're interested in learning more about human physiology, feel free to go and check out my playlist on that in my channel. Now, today's video is going to be just an introduction to the cell, some of the organelles, some of their functions, just to really get to know everything really deeply. And then we're going to continue on this playlist, kind of exploring their roles. If this video helps you, if you truly gain something from it, please consider liking and subscribing, and maybe leaving a comment. Okay, without further ado, let's get started. So, when we start talking about cells, okay, before we even dig into all of that, we have to briefly touch on the principle of cell theory, okay, which a guy named Theodor Schwann really came up with. And he basically said that the cell is the smallest structural and functional unit, which is capable of carrying out life processes. And the functional activities of each cell depends on the specific structural properties of that cell. And he also mentions that cells are the living building blocks of all plants or animal organisms, and it is the basic unit of life. And an organism's structure and function depends on the collective structural characteristics and even functional capabilities of the cell. And finally, because of this continuity of life, the cells of all organisms are fundamentally similar in structure and function. Okay, so this is just kind of the basics of the cell theory, okay? And I think it should be pretty intuitive to everybody. Now, when we talk about human body, we have about 30 trillion cells in the human body, and there are like approximately 200 different cell types, right? We can talk about cardiac cells, liver cells, kidney cells, skin cells, muscle cells, right? There are so many different subcategories of a cell. And, it's really just based on the variation in the structure and the function of each cell, okay? But despite all of those variations, all of these cells share many common features, okay? Such as being surrounded by this, protective layer called a plasma membrane, okay? Such as having this kind of watery gel-like substance inside called the cytosol, or even having this kind of brain of the cell, which is called the nucleus, okay? So they all share common features. Now, let's go ahead and talk about some of the things that you can find inside of a cell, okay? And let's go ahead and start out with this plasma membrane, okay? Here's that plasma membrane right here. And, we can go ahead and start labeling these. So right here, we can find ourselves a plasma membrane. Okay. Now, what does this plasma membrane actually look like? Well, if I take a plasma membrane portion, right, and I kind of zoom in on it, here's what I'm going to see, okay? So the plasma membrane, as it suggests in the name, it is a membrane, and it's basically a structure that encloses every cell in your body. And it's composed mainly of lipids, okay? Now, there's different kind of lipids it could be made up of, right? And for those of you who are new to this, lipids are just fat, okay? And these lipids, they will kind of assemble themselves in an orientation where they form a bilayer. A bilayer just means a double layer of these fats, okay, arranged in a very specific manner, which we'll take a look at, okay. And we'll have a whole video on the plasma membrane and some of its functions. But the plasma membrane, it functions as a barrier because it separates the inside of the cell, okay, from the outside of the cell, okay, or anything that is surrounding the cell. And it is also termed to be selectively permeable, meaning that it actually controls the movement of specific molecules into or out of the cell. Okay, so that's very important when we start talking about the plasma membrane. Now, I said that it is made up of lipids, but it can also contain other things like various channel proteins or carrier proteins, even some receptors. Again, we'll talk about this in the plasma membrane's own video. But the main type of lipid that you'll see is called a phospholipid. Okay, so this guy right here is called and let's switch this font a little bit. This dude here is called a phospholipid. Okay, now remember, lipid just means fat, and phospho means that it has this group on it called a phosphate group. Okay, alrighty. Now, if you're curious about the structure of lipids and the function of lipids, again, feel free to go and watch my videos. I have videos on structure and function of lipids later. But now let's take a look at the actual structure of this phospholipid. So, in a general sense, this guy is made up of two sides. Okay, and it looks like this. This portion and this portion. This portion we call a tail, and this portion we call a head. Okay, it's pretty easy to see, right? It's this giant circular thing that looks like a head. Often, though, we say that this head is actually polar. Okay, so it's a polar head, and because it is polar, it has this property where it's actually very hydrophilic. Okay, hydrophilic. Now, this is not a chemistry video, so I'm not going to talk about what polar is, okay? But just know that this basically has a kind of a negative charge, okay? This head here, it has a negative charge to it, okay? And it also is very polar, which means that it can interact with water, right? Because if you guys know, water molecules, right, they exhibit this characteristic that they're very polar. And if you've ever heard of the phrase like dissolves like, that should already tell you that this head piece of the phospholipid will be able to dissolve in water, okay? Or in other words, interact with water with no problems. Okay, great. Now, what does it mean to be hydrophilic? Hydrophilic is referring to the fact that this guy loves water, okay? And again, we said that it loves water because it's polar, okay? So, this guy's polar, and because it's polar, it can interact with water, and we give that a special name. We say that that molecule, or that this portion of this molecule, is hydrophilic. Okay, what about the other portions of this molecule? Okay, well, the other portions of this molecule are this tail. What is this tail made up of? This tail, guys, is made up of fatty acids, okay, which are a kind of lipid themselves, okay? So watch this. Here's a fatty acid, and here's one fatty acid that make up the tail. Now, they could be different, and usually they are. In a phospholipid, you can usually have a saturated fatty acid, and the other time you can also have an unsaturated fatty acid. Okay, and the only difference is that the unsaturated fatty acid has a double bond or covalent bond introduced in the hydrocarbon chain. Now, what are the properties of this tail that contains fats, or these fatty acids? Well, I want you guys to think about it critically, okay? If you've ever tried to mix water and oil, okay, you'll notice that they don't really dissolve with one another. In fact, one ends up being on the top and the other ends up being on the bottom. And that's because they kind of repel each other. And that is due to the inherent properties of these molecules, okay? So, these fatty acid tails, they are actually non-polar, okay, or uncharged, and they are hydrophobic, okay? They're hydrophobic, and that's actually the main reason why they cannot interact with the water. They don't like water, okay? So here, let's show a broken heart, okay? Because they don't like water. Look at that, okay? All right, so let me make this even simpler for you. Hydro means water, okay? Philic or philia means love, so hydrophilic means loving water. Phobic means fear, so this guy fears water, does not want to interact with the water, okay? This is how you remember which is which, okay? Now, there are examples I could give you of what this head specifically is, because I told you that these tails are specifically fatty acid molecules. Well, this head can sometimes, and it can differ, but you can attach many things to this head, and one common example is choline, but there's other examples. You can, for example, attach ethanolamine to there, and many other substances. Okay, but this is the main lipid that actually makes up that bilayer, that phospholipid bilayer membrane, okay? And if I take a whole bunch of this guy and assemble it together onto a membrane, here is what it would actually look like down here, okay? This is what it would look like. Now, we have to, using and understanding the properties of this single phospholipid applied to this whole membrane. So what do you notice? Let's say that this is the inside of the cell and this is the outside of the cell. You'll notice that these guys have oriented themselves in a very interesting manner where the polar heads that are hydrophilic are oriented to the sides that are facing water, okay? So the inside of the cell we know has some water, and the outside of the cell we know also has water. And this makes sense why these guys orient themselves in this way, because obviously you want these heads to be able to be dissolved in the water. But this means that this middle layer, okay, which is just a whole bunch of these fatty acid tails interacting with one another, okay, is then extremely hydrophobic, okay? This adds a level of selectivity to the cell, meaning that any molecule that is water soluble, okay, and therefore polar, or any molecule that's also charged, will not be able to enter this membrane, okay? And that's because of this hydrophobic layer, okay? So maybe it can interact with the polar head because it's water soluble, but then as soon as it gets close to this hydrophobic area, it gets repelled. So some examples of water soluble molecules are like glucose, okay? It'll try to get in, but it can't. It cannot readily penetrate this lipid bilayer. Other examples are ions, things like sodium. Sodium ions, they can't readily penetrate this plasma membrane, okay? They cannot do that. They require specific proteins that are spanning this membrane, such as channels or carrier proteins, to assist them. And we'll talk all about that in another video. But okay, now we can see that this membrane is selectively permeable, and it can even become more selectively permeable, and believe me, we'll talk about that later. One thing I would like to add, though, is that the membrane can be studied with these yellow molecules, which again we'll talk about, but this guy is cholesterol, okay? And cholesterol can be put into the membrane to affect the fluidity of the membrane. So for example, if there is high amounts of cholesterol, then the membrane fluidity is lowered, and the membrane is actually very rigid. That's what it's said to be. And what I mean by fluidity is the fact that these guys are not constant, okay? They don't just stand still. They twirl around, they switch places, they're constantly moving, and thus this membrane is constantly in a kind of like fluid mosaic, if you will, okay? And so the presence of this high amount of cholesterol can decrease that fluidity and make the membrane more kind of tight and rigid, okay? All right. So that's a little bit about the membrane of the cell, okay? We know that it encapsulates the cell. Great. Now let's talk about the brain of the cell, the nucleus, okay? And in this diagram, you can see the nucleus is actually this big brown structure almost in the center of the cell, okay? And so let's go ahead and write that down for ourselves, right? So here is the line leading up to it, the nucleus. Okay, now the nucleus like I said, it is the brain of the cell, okay? And the reason it's called the brain of the cell is because it houses the cell's genetic material, DNA. It houses DNA, right? And here it is. Here's that DNA. And we'll take a closer look at it in a second. But there's that DNA. And you guys know what DNA stands for, deoxyribonucleic acid. Okay? And you guys also know that DNA serves as a genetic blueprint during cell replication, right? So when the cell wants to split into two, right? The DNA has to be duplicated, and that means that each daughter cell gets a copy of that DNA, right? So everything is all good and dandy. We can carry on with our day without any problems. And this is important because the DNA has information inside of it that directs protein synthesis. And as you will go through this playlist, you'll see that proteins are extremely important. And I already have a video out on proteins, so if you want to know their functions, you can very easily go and watch that, and you'll figure out how important proteins are in your day-to-day functioning. So DNA extremely important as it helps make this proteins. Now, the nucleus, it has many regions, okay? Again, we won't get into it in this video, but for example, the nucleus itself is surrounded by this kind of double-layered membrane, okay? Which again is also phospholipids, right? Anytime you see a membrane, it most likely has phospholipids or other kind of lipids assembled in it, okay? But it is surrounded by a double-layered membrane, and it has these little pores that are called nuclear pores, studied in there, okay? Now, I told you that there are these regions, right, which contain the DNA, right? But it's not that simple. It's not that simple, and we actually have to zoom in on this to get a better kind of picture or idea of what I'm actually talking about. So let's take a big chunk of this guy and zoom in on it, okay? And you know what? Let's make this a little bit bigger so everybody can see what I'm talking about. Okay, so I'm sure in some sort of biology class, high school or whatever, you guys have come across a chromosome, okay, which is this kind of X-like looking thing. Now, this structure is the exact same as these structures over here. The only difference is that this guy is more compact and condensed. But what is it? Well, in order to understand what this is, we have to go way back. We have to come here. We start out, okay, as this DNA right here, okay? So look at this. Here is our DNA molecule. And DNA is just so long that we simply do not have enough space to store it inside of the cell if it was just kind of in this open, unwinded configuration, okay? But you can clearly see that the DNA already, it twists upon itself. It has a kind of double helix structure, okay? Now, this is good because it helps with the storage and the length, okay? And, it's obviously due to many things, such as hydrogen bonding between the bases. But regardless of the fact, making this thing twirled helps with storing it. But guess what? That's not enough. This guy is still pretty huge. How do we make sure that we can fit this giant amount of DNA inside of the cell? Well, guess what? We're going to twirl it, okay, and twist it along these groups of proteins in blue. Now, you might be asking me, what are these proteins called? Well, these proteins are called histone proteins, okay? And, if you want to get into it, technically this is an octamer. This is a complex of 8 proteins, okay? But, that's too much information for this video. Essentially, we take these DNA molecules, right, and we'll kind of wrap them around these guys, okay? If you're curious, it's about 1.65 turns, okay? That is the actual number, all right? It's approximately 1.65 turns this DNA wraps around these histone proteins, okay? And you'll see that as this guy will keep getting wrapped around it, right? What are we doing? We're essentially condensing this DNA so that we can store it, okay? And the combination of the histones and this actual DNA is called a nucleosome, okay? And we'll keep doing this until the DNA eventually starts looking something like this, where it's essentially just this line and it's studded around with these little blue dots, the blue dots being these proteins, okay? So at this point, we actually call these little lines now, we call them chromatin, but we call them chromatin fibers, okay? Because a fiber is usually just a strand of something, okay? In this case, it's a strand of these chromatins. Okay, now what ends up happening is that when the cell has to divide, meaning that, I have one cell, right? And this cell is, almost about to die. So guess what? It's going to replicate itself, okay? And it is going to pinch off in the middle and turn into two separate cells, okay? Two new cells. When this is happening, the genetic material inside of that one giant cell has to be duplicated. So it goes from one little twirly thing into two little twirly things, making this chromosome. And then when the cell splits, one of those twirly items will come to this side, okay? And one of those twirly things will come to this side. And that means that once the cell divides, each cell has now got a copy of the genetic material stored in it, okay, or of the DNA. Now, what is this little twirly thing called? This is a chromosome, and this is what the chromosome looks like during cell division. It looks very much so compact like this. Now, at the start, I told you that these little twirly shapes over here are the exact same thing as this chromosome here, but they're actually less condensed. So what would they be called? I'm so glad you asked. They're actually just this chromatin fiber. They are just studded around in this chromatin fiber shape, and whenever the cell has to divide, they become more condensed, and they turn into chromosomes. Okay? Great. That is the basics of the DNA. Now, what is so darn important about this DNA? Well, I'll tell you. If I have to zoom in on a DNA, okay, here it is. I'm zoomed in on it. I can say that a DNA molecule contains genes, okay? Let's say that this section of this actual DNA molecule is gene X. Now, what genes do is that they code for a specific sort of molecule, which is called a protein, okay? But you can't just turn a DNA into a protein directly. There has to be an intermediate step. So let me give you a breakdown. Here's what's going to happen. The cell will go ahead and read this portion of the DNA that is called the gene, and it'll produce a kind of intermediate molecule, right, which is also kind of wavy, and that one's called an RNA. So we had DNA turn into RNA. Now, DNA was deoxyribonucleic acid, RNA is ribonucleic acid, okay, and this guy is specifically called a mRNA. M stands for messenger. It's a messenger RNA. This messenger RNA will go and it'll interact with specific complexes that are available inside of the cell. They're really just these kind of like giant red shapes, okay, right here. They will read this RNA, and from this RNA, they will actually make a weird-looking structure called a protein. Okay, there we go. They'll make a protein. Now, what are these structures called? These guys are called ribosomes, and we'll talk about them in just a second. But this is how proteins are made. We go into the nucleus, which is the kind of brain of the cell. I like to think of it as a library, a giant source of information. And I go and look for a specific thing that I want. I want to make a specific protein, so I go to the portion of the library, the portion of the gene that contains that information. Then I create this messenger RNA, which takes kind of like a blueprint of what I need to create into these ribosomes, and these ribosomes will then make that for me, and then I have that functional protein. Okay? And that's what we're kind of seeing in this depiction over here. So if I were to zoom in right over here, guys, right over here, right here, I would see what I'm talking about right now. Here's that DNA. There's a specific protein that will go and sit on that DNA and read it, okay? And then it will make that mRNA. If you're curious, that's called an RNA polymerase. And it'll make mRNA. This mRNA will then exit the nucleus, okay, through that nuclear pore that we talked about. It will go and interact with ribosomes and create the protein. Now, you might be thinking, hey, what's this blue structure? And why is it close to this actual nucleus? Great question. You'll notice that we have these blue membrane-bound kind of organelles, okay? And these organelles are called the endoplasmic reticulum. Specifically, though, there are two types. These guys are called the rough endoplasmic reticulum. Okay, so the rough, I like to abbreviate it as ER. What does the endoplasmic reticulum do? The rough endoplasmic reticulum is studded with a whole bunch of ribosomes. These red guys right here, these are all ribosomes. And that's actually the reason why it's called a rough endoplasmic reticulum, because these ribosomes, they will come and attach themselves to the outer membranous surface of the endoplasmic reticulum, the rough endoplasmic reticulum, okay? And that's why it's called rough, because it looks rough. Now, what do these ribosomes do again? In our example, we said that these ribosomes, they actually create proteins. They synthesize proteins from that mRNA. That's great. That's great. So guess what? The rough endoplasmic reticulum, its function is to synthesize proteins because it has ribosomes attached to it. Okay? And you can see that the structure and where it is actually placed is not random at all. It is no mere coincidence that this rough endoplasmic reticulum is situated right outside of the nucleus, where as soon as this RNA leaves, it is very likely to encounter a ribosome attached to the endoplasmic reticulum and immediately start the process of protein synthesis. Okay, so it is by no coincidence that this happens. Now, once this protein is getting synthesized, right, it will be synthesized into the lumen of the actual endoplasmic reticulum. Okay, lumen just means the interior surface of something, all right? So it'll be synthesized into the lumen of the actual endoplasmic reticulum, the rough ER, and then the rough ER will kind of package it, okay? So like I said, it packages it, okay, and it folds that protein, okay, and then it pushes it into this, portion of the membrane that is kind of bulging out of the endoplasmic reticulum, and it secretes it out, okay? And this guy right here that it secretes out is called a vesicle, okay? It's just a membrane that contains that protein that we just made. Now, where does this guy go? Okay, well, this guy will follow it in a second, but he'll go on to another location called the Golgi Apparatus, and we'll talk about that later, okay? But for now, we have to mention this other kind of maroonish looking membrane structure that is just outside of the rough ER. Well, this guy is actually the other portion of the ER. He is called the smooth ER, okay? And it's smooth because there is no ribosomes attached to it, okay? So the smooth ER it likes to package a lot of the secretory products that we're about to send out, right? But really, it is the site of lipid synthesis. And there are certain lipids that are made in here. So, for example, phospholipids are made in the smooth ER. Cholesterol is made in the smooth ER. Okay. It is also responsible for other things. For example, calcium storage as well. Smooth ER does that. But those are the functions of the smooth ER, okay? And after it has made something, it will do the exact same thing as the rough ER. It will package it into a vesicle, okay? And then it will make that vesicle kind of bud off and then travel to where it needs to go to the cell, okay? Great. Now, let's talk about this Golgi over here, okay? Because this is important. Now, remember we said that we made this protein on the interior surface of the ER, then we packaged it into a vesicle, and then we pushed it out. Where does that guy go? Where does that guy go? Okay, well, that guy heads to the Golgi Apparatus, which is this guy right here, this pink guy. This Golgi Apparatus, right? It is going to receive the vesicle that is coming in. Okay, so imagine this is the vesicle containing our protein of interest. Okay, so boom, boom, boom. Here's our protein of interest. It receives it from the endoplasmic reticulum. Okay, and since, remember, that vesicle is just a portion of the membrane, it can easily just fuse with this other portion of this Golgi membrane. And what this Golgi is going to do is it will take those actual proteins or anything that the endoplasmic reticulum sends it, whether we're talking from the smooth ER or the rough ER, okay? It will modify them, package them, and distribute them, okay? So if we're talking about a protein, it'll distribute that newly synthesized protein to where it needs to go. If the protein needs to end up on, for example, the cell membrane, then it'll make sure it gets there. And how does it do that? Well, it puts on a little signal sequence on it that tells it, hey, you need to go to the membrane. Or it says, okay, you need to be in the cytosol. Or you need to go and be secreted outside of the cell. Right, so maybe it's a hormone. Okay, sometimes hormones are like that. So they will be packaged and modified, and then from the Golgi they'll be sent outside of the cell. Okay, so that is the job of the Golgi, and you can see how closely tied the function of the nucleus, the ER, and the Golgi are to one another. Right, it's really just very, very, very closely tied to one another. Their functions. Okay, now. What are some other things that we have to know? We have to know about this actual cell. And I kind of already mentioned one. It's the cytosol, okay? A lot of people confuse the cytosol and a cytoplasm, right? They think they're kind of interchangeable. But let me tell you this right now. The cytosol, okay, is the cell gel. Okay, it's like this kind of watery, gel-like substance, okay? And it is all over the cell. Now, the term cytosol specifically is the part of the cytoplasm that has no organelles. Okay? So the term cytoplasm it's going to include both organelles and the cytosol. But the cytosol is the portion of the cytoplasm without any organelle. All right? Great. But if you, again, want to be more particular, the cytoplasm is everything in the cell, excluding the nucleus, okay? Because the nucleus has its own thing. It's called a nucleoplasm, okay? But don't worry about that. Now, why do I mention this? The cytosol is a cell gel, right? It's kind of all over the place, literally all over the place. All of this kind of stuff, these organelles, they're all embedded into the cytosol, all right? What are some of the other things that are kind of embedded in the cytosol? There are intermediary metabolism enzymes. Okay, what the hell does that mean? metabolism, and we'll talk about it in a next video, where metabolism is the process by which our body likes to turn things from one thing to another, okay? Now, specifically, we can have metabolic pathways that make us energy or that use energy. One of the ways that our body makes energy is through a process of which we take glucose, okay, and we break it down in a series of 10 steps into a molecule called pyruvate. And in this process, right, which is, by the way, called glycolysis, we generate energy, okay? We generate energy in the form of ATP, adenosine triphosphate, okay? We'll talk about this a bit more later, but this process of glycolysis, okay, which is a metabolic pathway, occurs inside of the cytosol, okay? This all occurs right inside of the cytosol, and so the cytosol, when I say has intermediary metabolism enzymes, it has enzymes related to glycolysis. Another way we can make energy, and in fact we can make it very quickly, is by taking a molecule called phosphocreatine, okay, and a byproduct of ATP called ADP. So we have ATP, which is adenosine triphosphate. ADP is adenosine diphosphate. So it has two phosphates. And as soon as we kind of push these guys together, we can generate energy, so ATP, right? And we create creatine, okay? Now, how does this happen? Well, phosphocreatine had a phosphate. This guy had two phosphates, and it only needed one phosphate to become ATP. So we just transferred the phosphate from this guy to that guy, and we made creatine instead of phosphocreatine, and we turned the ADP into ATP. Okay? And this reaction is actually sped up by the presence of an enzyme, which is just a molecule that speeds up reactions, called creatine kinase. That is the name of that enzyme, okay? And you know, anything that ends with A's in biology is usually an enzyme. Okay, great. And this is a one-step reaction, whereas glycolysis was around 10 steps, okay? Alrighty. And we'll talk about glycolysis in the next video. But the cytosol includes these intermediary metabolism enzymes, okay? Great. It's also involved in the transport and secretion of these vesicles that we were talking about, and we'll show how. It also includes stored substances such as fat droplets and little bits of glycogen. Now, if you guys don't know, glycogen is just a stored format of glucose inside of the cell. So you might have like these little giant globs that are just floating around in the cytosol. Some of them might be called fat droplets. Other ones are glycogen. Glycogen is a storage form of glucose. Fat droplets are a storage form of fats. Why do we have these stored in the cytosol? Because if we need energy, these guys can readily be broken down and used for energy. Okay? Remember, the whole point of just eating food is to break down the nutrients in the food and use them as energy. And sometimes we can actually store them in the body for use later. Okay? And that's what we're doing. Great. Now, I feel like I have to talk about a few more of these organelles. Okay, so far we've talked about the nucleus, the rough ER, the smooth ER, the Golgi, the cytosol, okay? But we haven't talked about, for example, the lysosome, okay? And the lysosome is this yellow guy right over here, okay? We'll call it the lysosome. You can really think about this guy as kind of a garbage disposal system, okay? Because anytime we have something that is kind of old or worn out or it's toxic or kind of like garbage to us, we just deliver it to the lysosome, okay? And the lysosome, it has these enzymes in it. They're called hydrolytic enzymes, okay? And they're very destructive, man. The enzymes in the lysosome are very, very destructive. They degrade anything and everything, okay? They break down organic molecules, these hydrolytic enzymes, okay? And like I said, they're kind of like the garbage disposal or the digestive system of the cell. They literally destroy anything that we deliver to them, okay? Whether it's a worn out organelle or some sort of foreign substance, right? This guy immediately gets rid of those, okay? Great. And you can see those little red dots in it are those hydrolytic enzymes. Hydro means water, lytic means to destroy. So these enzymes are using water to destroy these organic molecules that we deliver to them, okay? Another kind of organelle inside of the cell that's involved in destruction or detoxification of the cell is these bad boys right here in purple. They are called peroxisomes, okay? Peroxisomes. I actually did a video recently that covers one of the functions of a peroxisome, and that video was alcohol metabolism. So peroxisomes are involved in alcohol metabolism, which means they can be found in a higher concentration in the liver, for example, okay? And these peroxisomes, again, they are membrane enclosed, just like pretty much every organelle that we've talked about so far, right? Like lysosomes, smooth ER, rough ER, nucleus, Golgi. These are all membrane enclosed, okay? And this peroxisome, it detoxifies various kind of like waste products in the cell and any foreign toxic compounds that can come into the cell. So for example, when we consume alcohol, right, after alcohol gets metabolized, it releases some pretty dangerous metabolites, and so this peroxisome kind of gets rid of it. It detoxifies those toxic compounds from the alcohol. And also it can detoxify various waste products in the cell. All right. Now, the only difference between this guy and the other guy, the lysosome, is that that one had hydrolytic enzymes. This guy has oxidative enzymes, meaning that they use oxygen to kind of break down or detoxify the molecules that we deliver to them. Okay, great. And you know, if you want to know, one of the enzymes that they have is actually called catalase. All right, which is really good at getting rid of these things called reactive oxygen species in the cell that can be very damaging to like DNA and stuff. Okay. The next organelle we'll talk about is the mitochondria, and believe me, we'll go into more detail on the mitochondria. And I think everybody knows what mitochondria is. It is the powerhouse of the cell, as it's very well known for, okay? Now, in cell biology, mitochondria does just more than produce energy, but that is one of its well-known functions. And something that is, you know, it might be fascinating for you guys to know is that the mitochondria, unlike being always described as this kind of rod-shaped or bean-shaped organelle, it can actually vary so much in its morphology, okay? So it can look something like this, or it can like be all like wavy and messed up, okay? It can look like it can have a reticulum almost, look like the kind of ER at times, okay? And it just really depends on which cell you're kind of looking at, okay? So that's my kind of take on the mitochondria there. But what does the mitochondria actually look like if I zoom in on it, okay? What is the kind of basic structure of the mitochondria? And that's a great question. Let's zoom in on it and find out. So here is one mitochondria. So let's zoom in on it and find out. Okay, so here's a zoomed in version of the mitochondria. You'll notice that the mitochondria, just like the nucleus, it also has a double membrane, okay? And it has this outer membrane, okay, which is actually called the outer mitochondrial membrane, okay? It also has a space that exists between the outer membrane and the inner membrane. So what is this guy called? This guy's called the inner membrane or inner mitochondrial membrane, okay? You'll see abbreviations of these as IMM and this one as OMM. But this space that exists between the outer mitochondrial membrane and the inner mitochondrial membrane is called the intermitochondrial, or sometimes called intermembrane space. And I think this is fairly intuitive, right? Inter means between, membrane, between membrane, space between membranes. Okay, so that's the intermembrane space. All right, what else? What are these kind of little like wavy things? These are called cristae. Okay, and they're called cristae for a reason. All right, these cristae are part of the inner membrane, but what you'll notice is that the inner membrane is a little different than the outer membrane in that it is kind of like folded up and all like wrinkly. Okay, the reason we've done this is to increase the surface area. Okay, so the inner membrane has these series of foldings that are called the cristae, and it's to increase surface area. And you might be thinking, increase surface area for what? And I'll answer that question actually right now. The mitochondria, right? It has this kind of inner portion, this inner core, if you will, right over here, where it also consists of this kind of like watery gel-like substance, but we don't call it cytosol, right? We call it the matrix, the mitochondrial matrix, all right? That's what that inside portion is called, okay? Maybe I should put an arrow here. This is called the mitochondrial matrix, okay? Now, within this mitochondrial matrix, there are a series of proteins and enzymes that go through a very important metabolic pathway, okay, that is kind of like a cycle, if you will. And we'll talk about this in the next video, but they go through what's known as the TCA or Krebs cycle, discovered by Hans Krebs, all right? And this guy right here is very important in making energy. In fact, it gives us these electron carrier molecules called NADH and FADH2s, okay? Again, we'll talk about these more in the next video in detail, and it also gives us a little tiny bit of energy. But really, the most important part of the mitochondria, okay, in the fact that we call it the powerhouse of the cell, is one, that it has this TCA cycle occurring inside of its matrix. The other is that it has these series of proteins that are studded along the cristae or the inner membrane. And that's why we have to make the inner membrane all folded upon itself to have this cristae shape, because the more surface area we have, the more of these bad boys we can actually fit on here. Now, you might be asking me, why would I want to fit more of these guys in here? And the reason is, these are the guys that give the mitochondria its name of powerhouse of the cell, okay? You have these protein complex called complex 1, complex 2, complex 3, and complex 4. And this other guy, which is commonly known as ATP synthase, sometimes called complex 5, which is the guy that's making the majority of the ATP inside of the cell. Okay? So this is why the mitochondria gets called the powerhouse of the cell, because of this electron transport chain. Okay? And that is exactly what it is called. It is called the electron transport chain. And we'll get into why it is called that in the next video. Okay, great. But now you guys know, mitochondria extracts kind of energy from nutrients in the food, such as glucose, and it transforms it into a usable energy for the cell in the form of ATP, okay? And it contains enzymes and proteins for the TCA cycle and the electron transport chain, and it even has enzymes and proteins for other metabolic pathways, such as for breaking down fatty acids, and that process would be called beta oxidation. Okay, great. Now let's talk about another organelle called this guy right over here, centrioles. Okay, this is cool, man. Centrioles are these kind of two rod-shaped looking things at right angles of one another. Okay, you can see it right over here, and I'm not even kidding. They exactly look like this. And the centrioles are found in this kind of bubble called the centrosome. Okay, now these guys, these centrioles, they are a pair of, like I said, cylindrical structures at right angles to one another. And these guys, they form and organize something called a microtubule. Okay? What the hell is a microtubule? Well, these long, like spindly looking things that you see are coming out of it, those are your microtubules. Okay? Now, in order for you to understand what a microtubule is, we have to talk about something else first. We have to talk about the cytoskeleton of the cell. Okay? And believe me, it's simpler than it sounds. All right? Just like how humans need skeleton to give themselves a kind of shape and not just be a pile of, gooey flesh on the floor, cells also require a skeleton to kind of give them a structure and a specific shape and also allow them to change shape and move around. The kind of stuff that gives that cell that ability is called the cytoskeleton. Okay? And there are three main cytoskeletal components, okay? And we'll briefly talk about them. One is called the microtubule, okay? Microtubule. The other one, guys, is called the intermediate filament, okay? And the last one is called microfilament. Do not confuse this with microtubule. Okay, so that's the three components of the cytoskeleton of the cell: the microtubules, the intermediate filaments, and the microfilaments. But what are microtubules? Where do they come from? And what is their function? So microtubules, they come from these pair of centrioles that are sitting inside of a centrosome, okay? And you can see them right here. Now, in this diagram, I've only depicted them going in one direction, but guys, they're going in all directions. They are literally all over the cell, okay? They are literally all over the cell. And the reason that they're all over the cell is because you can kind of think of them as the highway for the cell. They are literally the highway for the cell. Now, if we look at one of these guys, right, if I actually zoom in on one of them, I will actually find that they are just long, slender, and actually hollow tubes that are made up of specific proteins. So let's kind of zoom in on one of them. And here's what you'll see. In green, you're seeing the microtubule. Notice it's hollow, it is a tube shape, and it is made up of these alternating different proteins, okay? These proteins that it's made up of, by the way, are called tubulins, okay? And if you're curious, there's two types. There's an alpha type and there's a beta type, okay? Alrighty. Now, this microtubule, it has so many functions. It can maintain the asymmetric cell shape. It can allow for complex movements of the cell, okay? It allows the cell to take a specific form, all right? It is a big structural component of cilia and flagella. So I'm sure you guys have heard of flagella, right? So you guys know you have like these kind of sperm cells, right? If you're male, and these sperm cells, they have a tail, right? And the way that these sperm cells actually move is by rotating this tail. And the way this tail rotates around is actually very much so dependent on the organization of these microtubules in a specific format. Okay? So microtubules help in the movement of cells. Specifically, they are the main structural and functional component of flagella. Okay? And there's also cilia as well, which, if you've noticed, you have certain cells within your body that have like these kind of hair-like extensions coming out of them, and those are called cilia. You guys ever, felt inside of your nose, you have these nose hairs. Those are cilia, okay? They have a protective function by, for example, guiding debris and dust and stuff away from your respiratory tract in your nostrils, okay? They are also placed in other places in your body as well, okay? But again, it's all dependent on the microtubules, all right? The microtubules, not just that, they also position the cytoplasmic organelles. So they tell, for example, the Golgi where to be. They tell the endoplasmic reticulum, like, hey, you got to be exactly right outside of the nucleus so that whenever the mRNA comes out, it can immediately interact with your ribosomes and become into a protein. Okay? So again, it's really important, this microtubule, right? Because it not only gives the cell a shape, it allows it to maintain that shape. It is a structural and functional component of both cilia and flagella. It positions organelles. It also acts as a highway for transport of vesicles. So remember how I said that we'll package a protein and put it into a vesicle, and then we'll tell that vesicle to go into the Golgi, and then the Golgi will package it, modify it, and then put it into a vesicle, and then tell it to go outside of the cell or be put into the membrane. How does that vesicle keep moving around? Does it just randomly float? No. It gets moved around on the highway of the cell, the microtubule. And it also is moving around on these specific groups of proteins called motor proteins, okay? So, for example, this red guy right here, let's say it's containing a vesicle that has a specific protein in it, okay? And this protein needs to go to the membrane or, away from the nucleus. If that's the case, this motor protein called kinesin, okay, called kinesin, will actually grab on to that vesicle that holds our protein of interest, and it'll start kind of walking one leg after another on this microtubule highway. I'm serious. If you don't believe me, go and search up kinesin walking on microtubules. You'll see that it is actually just going to walk on these microtubules. Of course, that process requires energy, or ATP, but it does it, okay? And it will transport our protein molecule from the nucleus to the membrane. But what if I want to bring something back from the periphery of the cell? What if I want to go from the membrane to the nucleus, all right? What would that look like? Well, you need this other protein molecule, this motor protein called dynein. Okay, and dynein is responsible for going in this direction, whereas this guy's going that direction. Okay, and again, same thing. This guy will grab on to a vesicle that contains either a protein or, maybe a damaged organelle. Okay. So remember how I said sometimes lysosomes can degrade stuff in the cell? Well, sometimes we can have damaged mitochondria, and they can be put inside of a giant vesicle, and guess what? These motor proteins will just carry them towards a lysosome, and the lysosome will destroy that mitochondria. Okay? So that's one function that just regularly occurs in a cell. All right. And by the way, if you're curious, when dyneins move, it's actually called minus end retrograde transport. Retrograde meaning it's moving kind of backwards. And then kinesin, when it's moving, it's called plus end anterograde transport. Anterograde meaning it's going forward, okay? Again, you don't have to know that. It's just for those of you who want that extra information, here it is. Okay. Great. So now we know what the microtubule does. Now let's talk a little bit more about the kind of intermediate filaments, okay? Oh, but actually, before I forget, the last thing I promise, that microtubules do is, remember how I said during cell division, one of these chromosomes gets pulled to this side and one of these chromosomes get pulled to that side? Well, the thing that's actually doing the pulling is the microtubule. So this guy that is actually attaching to this side of the chromosome and pulling it apart, and this guy that's attaching to this side of the chromosome and pulling it to this side, that is the microtubule. So during cell division, these microtubules, they actually arrange themselves into specific molecules called spindle fibers. Okay? So just to kind of give you an example, remember how I said they're kind of just studded all over the place, which makes sense because they're the highway for the cell? Yeah, well, they will actually all just kind of come back inside, okay, into this actual centrioles. And then the centrioles will move to the either side of the cell, to the opposite poles of the cell, and they will start remaking those microtubules and then turning them into spindle fibers. And at the same time, it will start pulling the chromosomes apart and making the cell kind of pinch in two. Okay? Great. So that's the final function of these microtubules. You can see that they're extremely important. Okay? And not having them is very dangerous. Now, the last thing, well, not the last thing, but the second last thing is the intermediate filaments. And guys, these guys are very simple. They are just kind of irregular, like thread-like proteins, and I've depicted them in this kind of bluish color here. They're like studded all around the cell, and they're very, like I said, irregular, thread-like proteins. They help resist mechanical stress of the cell, okay? And later on, when we're talking about the membrane, we'll see that they're actually part of these things called desmosomes, okay? Which, again, also help resist mechanical tension or stress, okay? Yeah, so that's what the intermediate filaments are good for. And a good example of an intermediate filament is keratin, okay? Keratin is a good example of that. Microfilaments, they are the smallest out of this entire category, okay? So this guy is the largest. This guy's intermediate, so it's in the middle, and then microfilaments are the smallest. There are usually two categories. We say that there is actin, which is a microfilament, and then there's myosin. So if any of you have ever studied muscle physiology or muscle biology, you will have heard about actin and myosin, but they're also in other cells, okay? And these guys, within a muscle cell, they are what actually cause the contraction of a muscle, okay? And, I already have a kind of review video on that if you want to go watch. It's in this same playlist. So, yeah. Now, actin is the smaller kind of molecule as compared to myosin. Myosin is bigger, but they're both microfilaments, okay? They play a very big role in contractile systems in general. So, for example, when you have a cell that is trying to kind of move around, they're using these actin and myosin filaments, okay? You'll later on, when we talk about immunology in the same playlist, you'll see that white blood cells need to go through a kind of amoeboid motion to move and squeeze themselves out of the capillary bed. They're doing that using this actin microfilament, okay? Another thing is that actin also, and by extension the microfilaments, they play a role in that cell division process. So remember I said that the cell has to kind of split in half in the middle? Well, how do you usually like split things in half, right? Like say I have a bread. I have to kind of like apply a force and the thing kind of pinches in half from the middle almost, and then now I have two pieces of bread. Same thing when the cell has to split in the middle. Something has to kind of constrict this middle area and pinch. That thing is actin. Okay, actin forms this kind of contractile ring around this cell and makes it kind of split. All right, and so that's why we say that usually when we're talking about the contractile functions of the cell, we're thinking about these microfilaments like actin and myosin. Okay, and we'll talk about them in way more detail later, okay? So just to kind of write things down, we said that this blue lines that I've drawn here are the intermediate filaments, okay, which again help resist mechanical tension. In red, which are kind of all over the cell and give the cell like this kind of contractile thing, are going to be your actin filaments or your microfilaments if you're more interested. This circular thing over here is just like random vesicles, right, that are just on their way to either go this way or go that way, whatever, okay? You might be wondering what these little red dots are that I've drawn here, and if you're thinking they're ribosomes similar to this, you're absolutely correct. We have two types. We have free ribosomes that are just kind of floating, in the cytosol, and we have ribosomes that are attached to the endoplasmic reticulum. Okay, great. So that's that. Okay, guys, that is going to finish this video, which was an intro towards cell structure. Next video, we'll start talking about an intro to metabolism. We'll talk about glycolysis, the TCA cycle, and the electron transport chain in a kind of, again, basic introductory video. And yeah, so guys, if you learned something from this video, if you enjoyed it, if it truly helped you, please consider liking, consider subscribing to the channel. It really means a lot, your support, and I'll see you in the next video.